Okay, so introducing the structures of our respiratory system here on this first page. Give you a second here to write it out, and then we'll go through and read just the general introductory stuff. Okay, before we talk about the structures, just let's introduce functions of our respiratory system as a whole, which has kind of three different things listed here. Probably the number one, most important thing that it does is it provides for gas exchange, taking up oxygen from our lungs that can then be transported in our blood to our cells, which is something that we have to do every second of every day till the day we die. We can only go a few minutes without oxygen. And that's because we use oxygen in our cells to make ATP, which is our body's energy. Okay? And in that production of ATP, what we call cellular metabolism, we produce carbon dioxide that must be expelled, meaning that we need to move it to our lungs and push it out of the body through breathing. So doing the exact opposite of what we do with the oxygen. By doing that, we actually regulate our acid-base balance. Because, and it's not something I really get into in this class, but carbon dioxide reacts with water inside of us and creates an acid, which would throw off our body pH. And so by just breathing and using what we call a bicarbonate buffer system, our respiratory system helps regulate acid-base balance. Something very, very important. Okay, and then I've just got a set of different things listed here. For number 3, contains the receptors for the sense of smell. Talked about that before. We'll talk more about the nose here on the next page. The filtering of inspired air, production of sounds for vocalization, and the excretion of small amounts of water and heat. Now, the term respiratory and respiration are just the kind of same term, just different forms of it. And the term respiration refers to kind of 3 different things here. Ventilation of the lungs, what we would, just typically call breathing. The exchange of gases between our air and our blood, that takes place in our lungs, and then between our blood and our tissues, takes place in our tissues, like out in your muscles, out in your brain, that kind of stuff, which provides us the ability to use that oxygen, like I said, to make ATP in what we call cellular metabolism. Okay, so that's just more of the kind of physiology behind what our respiratory system does. Now let's talk about the structures that make it up. We can divide our respiratory system into what we call upper and lower tracts, which is kind of the anatomical way to divide it. The upper respiratory tract includes our nose, the nasal cavity, the divisions of the pharynx, naso-oro-laryngeal pharynx, and then various associated structures. I didn't include the mouth because your textbook doesn't. It really doesn't make sense to me because we use our mouth to breathe. More so, your mouth is important for digestion, but we do obviously breathe through our mouth. I guess it's maybe one of those associated structures. Whatever. Everything below that, starting at the larynx down, would be part of the lower respiratory tract. So your larynx, the trachea, all segments of what we call our bronchial tree, which is an air tube system within our lungs. And then obviously the lungs as well, that enclose those respiratory passageways. Now, this figure here that I've kind of pointed some stuff out on is from a different textbook. Kind of like it a little bit better than the one that's over on the right. This is the one from your textbook. Just a little more complete, I should say, as far as including things. I really like the fact that they bracketed out. Neither of these pictures will be on the exam, I promise you that. But I just wanted to kind of point some stuff out, so I included them. The other way to divide the respiratory system up is based on functionality or functionally into what we call a conducting zone and a respiratory zone. The conducting zone filters, warms, and moistens air, and then just conducts it into the lungs. So it's just airways that move it into the lungs from our nose down to what we call our bronchioles and terminal bronchioles, which are very distant branches or very tiny branches of our bronchial tree. And then I really like this picture because it zooms in at a little segment of our bronchial tree where our respiratory zone is. So it's showing up structures that make up our respiratory zone, tubes and tissues within our lungs, where gas exchange takes place. So this is where we bring oxygen into the blood and remove CO2 from the blood. Most notably, the structures that perform gas exchange are called alveoli, which is a plural form of alveolus. Alveoli I always compare to grapes, kind of circular structures. These are like little sacs inside our lungs. We have millions of them. They're in clusters, kind of like a grape, a cluster of alveoli. It's called an alveolar sac. At the top of them is an alveolar duct, kind of like the stem of a cluster of grapes. It's not a stem in that it's kind of holding it in place, but is actually more of an airway. So it's a tube carrying the air into the alveolar sac, and those branch off of what we call our respiratory bronchioles, which are microscopic airways. All this stuff is microscopic, which is why they've ballooned it up here in the picture. But like I said, neither picture will be on your exam. I just wanted to kind of point some stuff out. We've got better pictures to look at throughout the lecture. So we'll do that as we roll through the next set of slides. All right, we're going to start at the top with the nose, a structure we've talked about at various points in the semester, leading back all the way to when we talked about the skull bones. We learned about really more of the external nose, but a little bit about the internal nose as well. Actually, really, when I think about it, we've kind of probably talked more about the internal nose than the external nose. Regardless, the internal and external structures of our nose help to filter, warm, and moisten, as well as chemically examine the air that enters our body. First part of the nose, what we can see, portion visible on our face, consists of a supporting framework of bone and hyaline cartilage, covered with muscle and skin, and lined by mucous membrane, is our external nose. The bony framework is formed by our frontal bone, which kind of forms more of the superior portion. Remember, our frontal bone forms the forehead, but it also projects down enough to form a little bit of kind of the bridge of our nose. The majority of the bony framework comes from those two nasal bones, and then the kind of lateral aspects of the nose are formed by our two maxilla bones. Okay. So those are the bony framework. Our cartilaginous framework consists of septal nasal cartilage, which forms the anterior portion of our nasal septum. Remember, the nasal septum is something we've talked about before, divider between the left and right sides of our nose. The external nose, you can see it's a very thin plate of septal cartilage right down the center. Okay. On the outside, if you were to open up the nose, it would be that wall in between the two sides. On both sides of the septal cartilages, or septal cartilage, are the two lateral nasal cartilages. Probably figure out why we call them lateral cartilages, because they're lateral. They are inferior to the nasal bones. And then there's finally what we call alar cartilage, which kind of helps along with this last part of our external nose, forms kind of our nostrils. Okay, alar cartilage, let's see, helps form parts of the walls of the nostrils, and then the final structure of our external nose is composed of dense fibrous connective and adipose tissues. So it's just connective tissue with a little fat, and it's going to help to form that part that we can kind of flare. I do want you to know these different things that are labeled here, but I would also draw an arrow like you're pointing up the nose into it. The opening into our nose from the outside has two different names. I want you to know them both. Okay, nostril is probably the more common term. The term external nares is the other term. Okay, now since only one of them is labeled, you wouldn't label it with an E, you would label it with an I, nares. So external nares with an I instead of nares, which is plural. So draw an arrow up, label external nares / nostril. Over on the right is a frontal section where the face has essentially been removed. So we can see kind of deeper parts of the nose. In other words, we can see the parts of our internal nose, the large cavity beyond the nasal vestibule. I'll talk about what that is here in a minute. In the anterior space of our skull, lying inferior to our nasal bones and superior to our mouth, lined with muscle and a mucous membrane. So we just learned about the external nares. Let's talk now about what we call the internal nares, which are the openings that lead from our internal nose to what we call our pharynx, our throat. So this is the opening into our throat. We have got two of them because there's two sides of the nose. Internal nares or choani, which is a fun word to say, which we won't see those here, but if I zoom ahead into this sagittal section, we see a bracket showing this space here from the internal nose into the throat, labeled as an internal nares. Once again, I instead of E tells you it's singular, and we can only see half the skull there. So that's why it's listed that way. Ducts from our paranasal sinuses and the nasal lacrimal ducts also opened into the internal nose. We've talked about those before, and we'll look at them again on the next page. Our space within the internal nose is called the nasal cavity, and components include a nasal vestibule. This is the anterior portion just inside your nostrils. So this is where like the boogers are in most cases, unless you're digging deep. It's lined with skin, but really what's important here are these thick, coarse hairs, which help filter out large dust particles. They're very unattractive, and I always joke that for whatever reason, as guys get older, we grow them much longer and more grossly, but they're an important structure. All right, the nasal septum, as I've mentioned, is a vertical partition dividing the nasal cavity into the left and right sides. Anterior portion is composed of that septal cartilage that we were talking about, but deep in the nose, it transitions from being that cartilage to bone. Two predominant structures form our nasal septum: the perpendicular plate of our ethmoid bone, and then the inferior portion made up of a separate bone called the vomer. There's also a little bit of contributions coming from maxilla and the palatine bones, but the majority of it are those perpendicular plate and vomer bones. Okay. Inferiorly, the nasal cavity is bordered by both our hard and soft palate. Remember, those are going to form the roof of our mouth. Hard palate forms the anterior 2/3 of our roof, and then the back third is from the soft palate. Your hard palate is formed by the palatine and maxilla bones. For total, contribute to our hard palate. So we can see here in our picture at the bottom, the floor of our nasal cavity also is the roof of our oral cavity. Let's see. Laterally, the walls of our internal nose are formed by the ethmoid, maxillolacrimal, palatine, and inferior nasal concave bones. So a lot go into forming the sides. And then the ethmoid, by way of that cribriform plate, forms the roof of our nasal cavity. Deep within our nasal cavity are three folds of tissue formed by our skull bones. There are what we call the superior, the middle, and the inferior nasal concave. We have them on both sides. They project from the lateral aspect of the wall towards almost all the way to that nasal septum, and they create these little irregular channels for which that air has to pass through. So it's not just a wide open space as air passes into our throat. The spaces are called meatuses. You call them spaces, grooves, whatever you want to call them, channels. But what they do is cleanse, warm, and humidify the air that we are breathing in through our nose. And then you can't see them. We'd have to zoom in under a microscope to show, but those olfactory receptor cells, neurons, lie in a region of membrane lining our superior nasal concave and the adjacent septum, forming what we call the olfactory epithelium. We talked about those previously back when we learned about the sense of smell in our sensations lecture. There's a picture and a couple of slides where it's labeled, but it's not like something you can just open up the nose and identify without looking at it under a microscope. As far as this picture goes, don't worry about it. Just the one there on the left. Okay, we have one slide I'm going to color, which I will be coloring some structures that we have seen before, have seen and talked about before. So a good portion of this page, really all of this, is a review for you guys from various points in the semester. Talking here about some paranasal sinuses, which are these first four, and then some other things that are kind of tied in to the nasal cavity. Visible in the picture, so I included them. Really not the only reason why. As we learned previously, paranasal sinuses are air-containing spaces in our skull bones, open or draining into the nasal cavity. They're lined with a mucous membrane just like the inside of our nose. Function of them is to lighten the skull, but they also add a little timbre or complexity to our voice. And we'll color them now. Hopefully they're pretty easy for you guys since we've talked about them before. Color the frontals orange, that green can be for the sphenoid. We'll go yellow for the maxillaries. Let's see, how about this nice dark pink for the ethmoids. And then kind of only included them because they were showing up in a picture or pictures are the mastoids. Oh, those black. And then I'll go baby blue for not a sinus, but a duct from our eyes into the nose, the nasolacrimal duct. Okay, we've got three different images. This is an anterior view, pretty straightforward there. Another anterior view, but this is a frontal section, so kind of like what this previous page we saw, the face was removed, so we can kind of see deeper in to the nasal cavity, and then the lateral view. Okay, in your forehead, within the frontal bone are the two frontal sinuses just above our orbits, our eye sockets. Letter A in our pictures. Pretty good size pockets, as you can see in all three images. Okay, only visible in this bottom image is letter B, which is a sphenoid or sphenoidal sinus, a singular sinus within the sphenoid bone. If you remember back when we learned about the sphenoid bone, I told you guys it kind of has that butterfly shape to it. It's a linchpin bone forming a big portion of our cranial floor. If you were to kind of just, draw a line straight, like in the temporal region of your skull, and then like straight back from kind of right between your eyes, where those lines intersect will be kind of the location of this sinus. So basically, almost smack dab in the middle, a little bit more anterior in our skull. So kind of in the middle, front to back and side to side. So it's kind of misleading here. It kind of looks like it's on the side, but it's really deep within the skull. Okay. Maxillary sinuses are within your maxilla bones, your upper jaws. Hopefully you guys remember that. Pretty good size sinuses. If you feel like you get sinus pressure kind of more like in your cheek area below your eyes, these are probably the culprits where you might have an infection or something like clogged up. All right, and then our final kind of true paranasal sinuses, our letter C here at the bottom. Not a singular sinus, but more like pockets. We call the ethmoid sinuses or ethmoidal air cells. Collection of sinuses that drain into the nasal cavity, like we show here. These are the ones that always give me issues when I'm sick. Similar to those are what we call our mastoid air cells, which we mentioned back when we talked about the temporal bone, which is where these are located. They're called mastoid air cells. If you remember, this little bump behind your ear is called the mastoid process. You can see a little arrow pointing to the middle ear cavity. That's what they connect to, which your middle ear cavity is obviously not your nose. But remember, there's a connection from your middle ear to what we call our nasopharynx, which is called our auditory or eustachian tube. So your air cells, mastoid air cells, connect to your ear, which connects to your nasopharynx, which connects to your nose. So it's a kind of fourth connection to your nose. I'm really reaching there. But I really only included them because they were in the picture. We talked about them before. Along with this guy that I just colored, the nasolacrimal duct, which hopefully everybody remembers is what drains our tears from our eyes into our nasal cavity and is why our nose runs whenever we cry. Okay, hopefully nothing too crazy there for you. All right, moving to our pharynx, something I just mentioned a little bit about. As I said and talked about the nasopharynx. This is what we commonly call our throat, a shared tube, because it is going to carry not only air when we breathe in, but this is what we use to swallow food and drinks and move food or drink into the appropriate tube. So let's read a bit about it. Once again, it's commonly called our throat. It's a skeletal muscular funnel extending from the base of our skull to the level of the cricoid cartilage or larynx, which we'll talk about the larynx on the next page. Lies posterior to the nasal and oral cavity, superior to our larynx, and just anterior to our cervical vertebrae. As I mentioned, it's a common pathway for respiratory and digestive tracts because both food and air must pass through this structure before reaching its appropriate tube. Air must travel into the larynx. The food and beverages that we ingest go into the esophagus. Our pharynx also provides a resonating chamber for speech sounds and houses our tonsils. 3 anatomical divisions exist for our pharynx. The superior portion, as I just mentioned, is called the nasopharynx. It's posterior to the nose. Posterior to your oral cavity or your mouth is the oropharynx. And then the inferior-most is the laryngopharynx. So let's go through them individually, learning about them. Nasopharynx lies posterior to the nose, extends to the inferior portion of the soft palate. Your soft palate, remember, forms the posterior part of the roof of your mouth, shown here in red. So from here up, this area is the nasopharynx. 5 openings exist into the wall of the nasopharynx. 10 from these, there's going to be 2 from those internal nares from our inner nose, 2 from the eustachian tube, eustachian or auditory tube. Remember, is that tube coming from your middle ear? And then 1 opening that leads to the oropharynx. This is where we find those pharyngeal or adenoid tonsils, tonsil, because there's only 1, okay, which if you remember, we've seen this picture before. just as recently as our lymphatic lecture, I included this picture, but with all these lines removed. And then I drew arrows and labeled the different tonsils. I told you we'd see it again. Here we are seeing it again. Your nasopharynx is lined with pseudostratified ciliated columnar epithelium. Then the cilia here acts to move mucus down towards the inferior part of our pharynx. Speaking of, the inferior parts are the oropharynx and the laryngo. Oro is behind the mouth from the level of our soft palate to the level of our hyoid bone. Contains only one opening for the mouth, which we call our fauces. Okay, so this is the opening from our mouth into our throat that we swallow through. Okay, it looks really small. They're showing it with a bracket in the picture, but you have to keep in mind it would be a lot bigger than that. What they're doing is having the picture of the tongue, or in the picture the tongue is elevated, almost pressed up against the roof of the mouth. So it looks like, you know, we can only swallow something really small. We can't swallow huge things, but we can swallow decent-sized food and beverages. Your oropharynx functions in both respiration and digestion. I didn't list it, but really your nasopharynx is only going to function in respiration, breathing, because we aren't ingesting food and drink, at least I hope not, through your nose. Your oropharynx is lined with non-keratinized stratified squamous epithelium, protecting against the abrasive nature of food. And this is where the parapalatine and paralinguals are at, your tonsils we've previously learned about. The inferior-most portion of your pharynx is called the laryngopharynx, extends from the hyoid bone to the esophagus and passes food into our esophagus, which is posterior. It passes air into our larynx, which then travels down into our trachea on its way towards our lung, which is anterior. The lining of our laryngopharynx is non-keratinized stratified squamous epithelium, and another term for your laryngopharynx is the hypopharynx. Not that I'm really worried about you knowing it that way, more so just the laryngopharynx. Okay, obviously, you know, the picture is very important because we've got a lot of stuff going on that's pertaining to this lecture, but also our digestive lecture. As I mentioned, you know, the pharynx is a shared tube. So in this lecture, we'll talk about the pharynx again. I won't include the picture again, but as you can see, you kind of see some of the same parts in that picture where I do talk about the pharynx again. So a very important picture. I'll tell you right now, you're going to see it again next week. So highlight the following. On the left side of the picture, from sphenoidal sinus down to trachea. So everything except for sphenoid bone on the left side would be fair game.
From
Sphenoidal sinus all the way down to trachea. So everything but the top term, sphenoid bone, I'm not worried about you knowing. You know it already, right? Okay, on top, I want you to know the three nasal meatises, superior, middle, and inferior. Okay, here's the deal, though. Don't confuse the meatises, which are the little passageways, with the conchae. Look at the conchae. Those are the folds. Little grooves are the meatises. I want you to know both meatises and conchae, but you know, keep them straight. So those things on the top, know the frontal sinus. The olfactory epithelium is that area right there at the top of our nasal cavity we've mentioned before, where those olfactory receptor cells are located that allow us to have the sense of smell. Once again, know the three nasal conchae. The nasal vestibule is that space, like I said, where we find those hairs to help filter out large dust particles. Just inside the external nares, remember, that's the fancy term for nostril. If you want to write nostril down, you know, you can. I want you to know oral cavity, the soft palate, the lingual tonsil. Speaking of lingual, remember that always refers to tongue. I do want you to know tongue. I don't know why, but they label the tongue on the tongue. And then you can go down and highlight larynx, and that will be it. Now we're going to talk about these cartilages, the vestibular and vocal folds here on this page. But I'm not going to worry about you knowing them quite yet for the picture anyway. Kind of tough to tell them apart there in that picture, but we've got pictures of them here on this page to show off the parts of the larynx. Which includes these cartilages and some more here after we get through these cricoid cartilages. All right, nickname for your larynx is your voice box. It lies between the root of our tongue and the upper end of the trachea, just below and in front of the oropharynx. It consists largely of cartilages attached to one another and to surrounding structures by muscles or more cartilage. It's considered part of our airway lungs and is lined with a ciliated mucous membrane, helping to remove dust particles. We call it the voice box because it's our organ of voice production. Our voice box by vibrating what we call our vocal folds, producing the sound. Now, sound originates down in your larynx, but the size, shape of our nose, mouth, and pharynx, as well as the bony air sinuses, help determine the characteristics of our voice. More importantly for speaking would be our mouth and the structure inside known as our tongue. It is impossible to speak coherent words without a nice usable tongue. Try it on your own. You'll sound like, Charlie Brown's teacher. Non-cartilages form the framework of our larynx and includes the epiglottis. The epiglottis is the only thing that you had to highlight as part of the larynx other than just the larynx as a whole. The epiglottis in this case is allowing for air to enter our larynx and then down into the trachea. So this would be when a person's breathing. Our epiglottis is a really cool structure. It covers this opening to our larynx. It's not labeled, but this would be what we call the glottis. And it functions like a trap door where it can hinge open like it is right now or close like what we do when we swallow. Okay. So it's a flexible flap of tissue, guards the superior opening of our larynx, and covering what we call the glottis, the upper region of the larynx, during swallowing to prevent entrance of food or drink into the trachea. Two parts of the epiglottis exist. The upper flaring, more large portion is the leaf. The inferior portion is called the stem. The stem is the tapered inferior portion attached to the anterior rim of the thyroid cartilage. The leaf is the broad superior portion that is unattached, and that's what moves up and down like a trap door, as I mentioned. Your thyroid cartilage is the largest of the cartilages and is known more commonly as the Adam's apple. It is going to cover the anterior and lateral aspects of the larynx, functions to protect the front of our larynx. It's more projected forward in men due to the influence of male sex hormones. A lot of testosterone influence there. A ligament known as the thyrohyoid membrane connects our thyroid cartilage to your hyoid bone. Just inferior to our thyroid cartilage is that thyroid gland, which is, I don't know, chicken or egg, which one was named first, but now you can see why this is the thyroid cartilage and this one's the thyroid gland. Now, in that anterior view, we have a hard time seeing the next cartilage. See it a lot better here in the posterior view, our cricoid cartilage. Ring of hyaline cartilage forming the inferior wall of our larynx. Happens to be the landmark for making an emergency airway in a procedure called a tracheotomy. And is connected to the first ring of tracheocartilage by what we call a cricotracheal ligament. It's connected to the thyroid by what we call a cricothyroid ligament. Like I said, it's kind of harder to show here in our cartoon version. Down here we've got a cadaver version anterior view where we can see the cricoid nicely both anteriorly and posteriorly. Together, the cricoid and the thyroid essentially constitute the box of our voice box. Now, in our posterior view or in the posterior view, we will be able to see the next 4 pairs, or I should say 2 pairs, 4 cartilages, the arytenoids, and the corniculates. Very small cartilages. The arytenoids, there's a pair of each, are these kind of bigger ones. Let me zoom in and show you. So the arytenoids are kind of this triangle that doesn't come quite to a point. At the very tip of those are very small, tiny, what we call corniculates. Okay, let's learn about them here. The arytenoids are posterior to the thyroid cartilage and are attachment site for what we call our vocal folds. Okay, which we happen to show here at the bottom as our true vocal cords. They contain elastic ligaments that vibrate when skeletal muscles move them into the path of outgoing air, which produces our sounds. The corniculate cartilages are horn-shaped pieces of elastic cartilage at the apex of each arytenoid cartilage. And you can see them nicely here and over here. Over here is a sagittal section, so the larynx has been cut in half from front to back. And I included this picture really to show the final portions of our larynx, what we call the cuneiform cartilages. We only show one because there's only half of the larynx there, but there are 2 total, a pair of them. And they are elastic cartilages anterior to the corniculates, supporting our vocal folds and the lateral aspects of the epiglottis. Okay. The mucous membranes of our larynx form 2 folds, or 2 pairs of folds. Our superior pair is called the vestibular folds, also known as false vocal cords. The inferior pair are the true vocal cords, also known as your vocal folds. If we go back to the previous page, those were labeled here. Like I said, it's kind of tough to tell them apart. Really, you know, it's tough to tell all this stuff apart from one another, except for the epiglottis, which is the only one I wanted you to kind of know independently. Just everything else together is, you know, in this picture, the larynx. As far as these 2 pictures go, these are the ones that I want you to know for the exam. What is already bolded for you? So epiglottis leaf stem, corniculate cartilage, thyroid cartilage, arytenoid cartilage, cricothyroid ligament, cricoid cartilage. Those are the things I want you to know, but I would also highlight the thyrohyoid membrane. Anything else is not important to know, though. And then I'm not going to worry about the sagittal section picture. It's kind of tough telling things apart. It's not my favorite, so I won't worry about it. Just these 2 will be what I want you to know. All right, inferior to your larynx, we have another nicknamed organ, our trachea, which we commonly call our windpipe. Carries air down to our bronchial tree. And we'll get into the parts of the bronchial tree here in just a minute. First, I'll let you write it out. Okay. Once again, our trachea is known as our windpipe. It's a flexible tubular structure just beneath the larynx, approximately 12 centimeters to 5 inches in length. Obviously, that's dependent upon how tall we are and how long that will be. 2.5 centimeters in diameter, that really doesn't change, which is right at an inch. It extends from our larynx to the superior border of our 5th thoracic vertebrae, where it forks, forming a right and left primary or main bronchus. Those are the very beginnings of our bronchial tree. Location is anterior to the esophagus, and it's made up of stacked rings of cartilage with fibrous spaces. The tracheal wall is 4 layers thick. It includes a mucosa, submucosa, hyaline cartilage layer, and then an adventitial layer. Might not be a bad idea, maybe over here in the little space. This is superficial, this is deep. So we're going to work deep to superficial. So you just keep your head straight on the order in which the layers are put together. This is the innermost or deepest. This is the outermost or most superficial. The mucosa, once again, is the inner layer. It's composed of an epithelial layer of pseudostratified ciliated columnar epithelium. Remember, pseudo looks like it's multiple layered, but it's not. Ciliated, it's got cilia. And there's an underlying layer of lamina propria that's connective tissue with elastic and reticular fibers. So it's got ciliated cells, but it's also got mucus-producing goblet cells that release the mucus to act like a glue trap to collect dust and other particles that we breathe in. The cilia on the ciliated cells are going to move that mucus and the dust that's trapped to it up toward the pharynx. At this point, we're trying to get stuff out of the lungs or away from the lungs, so we move it upwards into the throat, which means we have kind of two choices. We're either going to kind of like cough and spit that gunky, mucusy stuff up and out, or what happens probably more often is we actually just end up moving that mucus and the dust stuck to it down into our esophagus to carry it into our stomach, where it's then digested. It sounds really gross, but it's actually super effective. Surrounding the mucosa is a submucosa, a layer of areolar connective tissue, which is, like I said, surrounding the mucosa. The third layer is where you find hyaline cartilage, 16 to 20 C-shaped rings of hyaline cartilage wrapping around the submucosa. We all know what a C looks like. There's one. We can actually see from a cadaver one of those tracheal rings. You can see the nice C shape to it, but the open space of the C is not void of anything. There's something there connecting the two parts. This is what we call a fibromuscular membrane, where a muscle known as the trachealis muscle is located. That muscle can contract, which is going to decrease the size of this lumen, which means less air will pass through the trachea. It can also relax and make it more open. Now, it's not going to do drastic changes to the lumen, and what it will not do is allow the trachea to completely collapse. Now, your esophagus, which we show here, can absolutely collapse. Okay, it can shut pretty much completely down, and that's because it does not have the cartilage. It's just made up of muscle. The cartilage of the trachea is very important to prevent the trachea from collapsing. We don't want our airways to ever collapse when we're breathing. Okay, otherwise we can't breathe. Okay. The final layer of your trachea is called the adventitia. It's the outermost layer of areolar connective tissue, just like we see with a lot of outer layers of stuff. Just kind of helps hold the trachea, in this case, to surrounding tissues, holding it in place. Okay, let's talk about the bronchial tree, which is a series of branching air tubes within our lungs, beginning at the inferior portion of our trachea, and helps to divide our chest into right and left structures, and carries the gases into our right and left lungs. Begins with the two primary or main bronchi. Now, bronchi with an I at the end is plural. If you end it with bronchus, US, it's singular. Okay. Those are the first branches of our bronchial tree. They are the largest branches as well, coming off of our trachea and carrying the air into the right lung if we're looking at the right, and then the left lung if we're looking at the left. Okay. Those then branch and form what we call our secondary or lobar bronchi. And we call them lobar bronchi because they are associated with each lobe of our right and left lung. I guess there's two those there. You can cross one of them off because I don't need two. Your right lung has 3 lobar bronchi, a right superior, a middle lobar, and a inferior lobar bronchi. That's because your right lung has 3 lobes. Your left lung only has 2 lobar bronchi. Okay, and that's because it's only got 2 lobes. The 2 lobar bronchi are the left superior and inferior lobar bronchi. Your left lung is smaller than your right lung, and we'll talk more about this here in just a couple slides. But it's because of this notch that exists in our left lung called the cardiac notch, which accommodates what? What does cardiac always refer to? Your heart. Okay. Very good. Remember, your heart sits at that angle, points more to the left side of our chest and sits more on the left side of our chest. So our lung has to actually have an indentation out for that, which is why our left lung only has 2 lobes, okay? And therefore only has 2 lobar bronchi. Coming off of those, branching from them are the tertiary or segmental bronchi. We call them segmental because they are going to feed the different bronchopulmonary segments of our right and left lung, and are named as such, which I'm not going to really get into this stuff. This is when the lungs are getting real small and the bronchi are getting real small as well. Branching off of those tertiary or segmental bronchi are bronchioles, which are surrounded by smooth muscle, pretty small in diameter, millimeter or less, but still visible with the naked eye. A millimeter is small, but it's not microscopic. Our terminal bronchioles are the final branches of our bronchial tree, also not microscopic. We can see a 0.5 millimeter in diameter with a naked eye. But your microscopic branches coming off of those are called respiratory bronchioles. Microscopic being that we can't see them with the naked eye. And if I go back to that first page, where we talked about the respiratory zone, listed here as part of our respiratory zone are respiratory bronchioles, because they are part of that respiratory zone where gas exchange takes place. Remember, gas exchange takes place specifically with the structures called alveoli that we'll talk about more on that next, or this next slide. As the branching becomes more extensive in our bronchial tree, several structural changes occur. Our mucous membrane in the bronchial tree changes from pseudostratified ciliated columnar epithelium in our primary, secondary, tertiary bronchi to ciliated simple columnar epithelium with some goblet cells. Remember, goblet cells produce mucus. In our larger bronchioles to mostly ciliated simple cuboidal epithelium with no goblet cells. In our smaller bronchioles to mostly non-ciliated simple cuboidal epithelium. In our terminal. What that essentially boils down to is this. The bigger the bronchial structure, the bigger the branch, the more cilia and mucus it's going to have. Remember, mucus helps trap that gunk that we breathe in and move it up by way of the cilia. But hopefully, the farther in you get, the more branching you get. We've already trapped it because the farther in, we have less mucus-producing cells and less ciliated cells. Finally, we just have none in our terminal bronchioles. Plates of cartilage are gradually replaced by incomplete rings of cartilage in our primary bronchi before completely disappearing in our more distal bronchioles, which allows for more smooth muscle to increase, which allows for the ability to alter the lumen size through what we call bronchoconstriction or bronchodilation, where we can contract or relax the muscle to increase air or decrease airflow in and out of the lungs. You can't really change the lumen a whole lot, and it's like what I was talking about with the trachea. When the cartilage is there, it's harder to close or more open that airway than it is like the esophagus, which just has muscle. And the same thing we're looking at with the very tippy branches, small bronchial tree structures. Now, I do like this picture a lot. You can see a lot of things to it. It's something I just want you to highlight a couple things in, though. At the top, I want you to highlight the larynx. We just talked about that. The trachea is obviously nice and visible. As far as the bronchial tree, though, I just want you to highlight the right main and the left main bronchus. And then for the lung, the left lung, highlight the cardiac notch. Really, the other stuff's kind of just too tough to tell apart, especially the little branches. I do recommend, though, that you highlight or star this little box here showing you the branching, kind of a nice way to keep your head straight. And it would not be a bad idea. I would strongly recommend that you do this where it says main, put in parentheses primary. Where it says lobar, put secondary. Where it says segmental, put tertiary. Because they have two different names. I want you to know both names. For whatever reason, the textbook, like the previous edition, kind of went more primary, secondary, tertiary. And then when they updated the book, they are now kind of pushing you to know it as main, lobar, and segmental. My advice, it's just easier if you know them both, because you might see it both ways. At the very bottom right is essentially the same as what you're looking at here, the exception being the lungs have been removed. And in doing so, a lot of the very small branches are gone. But it's pretty cool to see what a cadaver bronchial tree all the way back up to the epiglottis as part of the larynx looks like. You can see the branching. It's pretty cool. Obviously, it's going to get a lot smaller than that, but you know, it's just probably too difficult to keep those things intact when they remove the lungs. Okay, and on this page, as promised, I now want to talk more about the primary structure of the gas exchange, what we call our alveoli, and their anatomical makeup. Okay, alveoli, as I said, primary structures of gas exchange of our respiratory tract, performing what we call pulmonary or external respiration, exchanging carbon dioxide that leaves the blood and oxygen that must enter the blood. Associated with alveoli are those alveolar ducts, alveolar sacs that we mentioned on that first page. The ducts are the passages that lead from the respiratory bronchioles into the alveolar sacs. Alveolar sacs are the clumps of alveoli having a common opening from those alveolar ducts. Each alveolus is only one cell thick, very similar to the pulmonary capillaries that surround them, which, as it says, also are one cell thick. This illustration here shows, you know, those different things I just mentioned. The sac is the cluster. The individual ball-like structures, like the grapes, are the alveoli. Surrounding each one of those, they don't show it over here for these three, but they do really nicely here, is a very dense network of pulmonary capillaries, which are the routes in which we are going to pull the oxygen into the blood and remove the CO2 from the blood. The ducts that are like the stems of our grape cluster are shown here at various points of our picture, and those branch off of our respiratory bronchioles, which we just talked about on the previous page, branching off of our terminal bronchioles. Down here at the bottom of the page is an illustration showing you a sectioned open alveolus to show its very thin wall. Like I said, is only one cell thick, enabling the gases of CO2 and O2 to pass very easily. CO2 passes into the alveolar lumen from the blood, whereas O2 passes the opposite direction from the alveolar lumen into the pulmonary capillary blood bed and then into the blood. So what this enables is gas exchange to occur with very little resistance, because the walls are so thin. Like I was talking about this in phys the other day, it's like 0.0005 millimeters, which is essentially like nothing. The surface area of each lung is increased because we have millions of alveoli in each lung, meaning we have a lot of space in our lungs for gas exchange to take place, which is a great thing. We absolutely want gas exchange to take place in high numbers. All right, I want to talk about this picture. The wall of a alveolus is predominantly formed by cells called type 1 cells. Type 1 alveolar cells, as you can see, are basically just like the cells that form the wall of a capillary. Very thin, plate-like, and they are one of the 4 layers that form what we call our respiratory membrane. The respiratory membrane includes these 4 layers. We're zooming in at this section of our picture to show them. The type 1 cells are 1 layer. Directly on the outside of that is what we call the epithelial basement membrane, which is just like the basement membrane of our capillary wall, which, if you remember back from our blood vessels lecture, the very last lecture we've talked about, we learned that capillaries were only made up of 2 things. It's endothelium and then a basement membrane. So those 4 layers constitute what we call the respiratory membrane, the barrier for which gases have to be exchanged. Now, it sounds thick, like I said. Those 4 layers really are nothing like 0.00005 millimeters. Okay. The other types of cells that you find in alveoli are type 2 cells, also known as septal cells. You can see here pictured, as well as down here in blue. Those produce a chemical that is found on the inside of each alveolar lumen called surfactants, which reduces surface tension, fancy way to describe force of attraction between water molecules of the fluid. More importantly, it prevents each alveolus from collapsing and sticking together as we breathe, which enables oxygen to be diffusing more easily. So surfactants important, produced by type 2 cells. There's also alveolar macrophages or dust cells, which are just like macrophages we've learned about previously in other places of the body. Phagocytes that are like Pac-Man chewing up, at this point, should be really fine dust particles and other debris that weren't filtered already. There's also fibroblasts producing reticular elastic fibers, which are labeled here in our picture. Not the fibroblasts, but the fibers anyway. Okay, as far as the different pictures, these pictures here on the top right and bottom left are showing you actual microscopic views of these different structures, but these 2 cartoon versions are the ones I want you to know. Highlight the following in this picture. I'll just start at the top with the terminal bronchiole, the respiratory bronchiole, alveoli, alveolar ducts, alveolar sac, and alveoli. Basically, this little box is a continuation of this box. So it might not be a bad idea to highlight that box so you keep your head straight on the direction in which air flows. I do also want you to know, like I said, this picture, everything that's already capitalized for you is what I want you to know. So the type 2 alveolar cell, the respiratory membrane that's circled out right here, type 1 alveolar cell, alveolar macrophage, and then the 4 components of the respiratory membrane that's zoomed in on this little box. Capillary endothelium, capillary basement membrane, epithelial basement membrane, type 1 alveolar cell. Okay, so now we've covered all the little tiny structures within our lungs. We haven't really talked about the lungs themselves, the organs that enclose those tiny structures. All right, let's talk about the lungs. Cone-shaped organs with each consisting of a broad concave base that rests on our diaphragm and a blunt peak called the apex projecting slightly above the clavicle. We have got several different views here. These are lateral views, left and right lungs, and then medial views looking from kind of the inside out. Right lung, left lung again. Top of the lung is the apex, comes to a point just above our clavicle. The base is the widest part, sits on top of our diaphragm. These are actually cadaver lungs. Pretty cool to see over on the right side of the page. Key thing to keep in mind about the lungs, it's not, and I have it capitalized, a hollow balloon-like structure. It's more like a sponge. And it does consist of those balloons that we have that we just talked about called alveoli, served by many, many pulmonary blood vessels. Our lungs lie against our ribs both anteriorly and posteriorly because our ribs, remember, wrap around from front to back. The surface of the lung lying against the ribs is called the costal surface. Remember, costal always refers to ribs. We can actually see here in our real lungs, the kind of grooves from those ribs. We call those costal impressions, or as I said, costal surface. The mediastinal or medial surface of each lung contains a hilum, which is not the first time we've seen an organ with a hilum. Hilum is a indentation. We talked about the hilum of a lymph node not too long ago. It contains primary bronchi, or this is where primary bronchi, pulmonary blood vessels, lymphatic vessels, and nerves enter and exit each lung. We can see them only here in our medial view. Hilum is where you find what we call the root, okay, which is how we hold or let me just read it. They're held together by the pleura and connective tissue, what we call the root of the lung. We talked about this just a couple of slides ago. Our left lung has that cardiac notch, concave portion where the heart lies. You can see it just a little bit here in our lateral view. It's got a nice view here in our medial view. But that's, once again, because your heart sits more on the left side of your chest. Each lung is divided into lobes and then subdivided into functional units called bronchopulmonary segments, which I'm not going to get into the bronchopulmonary segments. But I will talk about the lobes. Each segment is served by a tertiary bronchiole or a segmental bronchiole, as we learned. We then divide those bronchopulmonary segments into lobules. I'm not going to get into that. Now we're going to be really, really, really small. Those are served by our terminal bronchioles. As we learned previously, our left lung has only two lobes, a superior lobe and an inferior lobe, separated by what we call oblique fissure. Whereas your right lung has three lobes, a superior lobe, a middle lobe, and an inferior lobe, and there's two fissures, a horizontal and then an oblique fissure to separate them out. Each lung, and we've talked about these before, is covered superficially by a structure known as the pleura, a strong connective tissue, which is actually a double membrane. We introduced these back at the very beginning of the semester in our first lecture. We recently talked about the parietal and visceral pericardium. Kind of the same situation exists around our lungs. We have what we call the pulmonary or visceral pleura, which is the deeper layer covering the lung itself, whereas the parietal pleura is the superficial layer that lines the wall of the thoracic cavity. Now, between those two membranes is and it's listed here as a potential space. I like that term because it's really not a big cavity, because these membranes like to stick to one another. Okay, I always think of them like, when you see in movies and stuff when kids get their hands glued together. Okay, they're not literally glued together, but they have an attraction to one another because of the pleural fluid that's in this cavity, which creates a vacuum and helps them stick to one another. It also reduces the friction between the two membranes, which is very important so they don't, wear holes in them and stuff like that. Okay. We want this cavity to stay very small and essentially non-existent, since the membranes like to stick to one another. But a collapsed lung can occur, as you've probably heard of. There's a condition called a pneumothorax, which is where air gets in that pleural space. There's also what we call a hemothorax. I didn't list that off, but that's where blood gets in that space. Very, very dangerous situations. Okay. Now, as far as the pictures go, I'm not going to worry about, the real picture, which is cool, but, kind of tougher to tell things apart. I do want you to know these 4 images, though. And really looking at it, everything I would say would be fair game. So we talked about each of those different things, the apex, the base, the fissures, the lobes, the notch. What you should also make sure you can just do is, look at this picture and say, okay, that's definitely a left lung. This is a right lung. So you can tell that right away because of the number of lobes, which relates to the number of fissures. So, highlight, might not be a bad idea, right lung, left lung, right lung, left lung. A little tougher to tell things apart here in the medial views. And once again, key thing though, you can see 2 fissures here, only 1 here. Cardiac notch is only in the left lung, not in the right. All right, our final slide here is really not anything brand new, and it relates back to the last system we talked about, our cardiovascular system, more so relating it back to what we talked about when we talked about the flow of blood in and out of the heart. As we know, deoxygenated blood or high carbon dioxide containing blood returns to our right atrium, where it's then going to be pushed out of the right ventricle to travel to our lungs, where gas exchange takes place, which we've been talking a little bit about. Structure is called alveoli, where CO2 exits the blood, O2 enters the blood. Blood then is going to be rich with that oxygen, coming back to our left atrium and then be pumped out by our left ventricle to our systemic circulation. Just like our heart, which had a set of coronary arteries and veins, our lungs, our bronchi, and our trachea need their own arteries and veins. We call them bronchial arteries, bronchial veins, respectively, because the gases inside of the trachea, are not going to be exchanged as they move through. Picture's not important. Really, I'm not going to, I already asked you to trace the flow of blood through the heart and that kind of stuff. So really nothing specific too much to worry about here. Sound good?